Welcome to the deep dive. Today we're getting into something really fundamental about depression.
We are.
How it actually changes our brains. Specifically, its amazing ability to adapt and learn. That's neuroplasticity, right?
Exactly, neuroplasticity. And we'll also look at the positive side, how treatments can actually help the brain heal.
Yeah, that's the hopeful part. So we'll be zooming in on some key brain areas, the ones important for emotions, learning, memory.
Precisely. We'll see how depression affects them. But really, the core message today is hope. Our brains aren't fixed. They can change. They can recover. It's pretty incredible.
Okay. And to understand this, we looked at a bunch of scientific articles, stuff about how depression impacts this brain flexibility, and crucially, how different treatments can help the brain, well, essentially rewire itself. So the mission for this deep dive is to really get a handle on the biology behind these brain changes in depression and, the potential for recovery through these different interventions.
I'm good.
Right. So let's start with the basics. What exactly is neuroplasticity?
Okay, so think of neuroplasticity as your brain's lifelong capacity to change itself.
Lifelong.
Yeah, structurally and functionally. It does this in response to, well, pretty much anything new experiences, learning something new, even an injury. It's how we adapt.
Like learning guitar or something.
Exactly. Or navigating a new city. And this isn't just happening at a large scale, like entire brain regions changing. It's happening right down at the level of individual brain cells, neurons, and the glial cells that support them.
Glial cells, like support crew.
Sort of, yeah.
Huh.
And it even happens at the connections between neurons, the synapses, tiny, tiny changes that add up.
So this isn't just something that happens when you're a kid?
Oh, definitely not. I mean, it's really ramped up during development for sure. But neuroplasticity is with us our entire lives. It's absolutely key for adult learning, memory, and like I said, recovery when things go wrong.
And that brings us to depression, because we see changes in this adaptability there, right?
We do. Pretty significant ones, actually.
Okay, so how does depression actually, interfere with this? I know some studies use MRI scans to look at the physical size of brain areas.
That's right. MRI studies have pretty consistently shown that certain brain regions tend to be smaller in volume in people with major depressive disorder, or MDD.
Which regions are we talking about?
Primarily areas really involved in emotion, learning, and memory. Specifically, the hippocampus and the amygdala often come up.
Okay, hippocampus, that's the memory center, kind of. And the amygdala is more the emotion hub.
You got it. Hippocampus is crucial for forming new memories, and the amygdala is like the brain's alarm system, especially for negative emotions. But the changes don't stop there.
No.
No. Researchers also see thinning of the cortex, that's the brain's outer layer, in parts of the prefrontal cortex.
The thinking part of the brain.
Exactly. Specifically the orbital and medial areas, which handle things like decision-making and managing emotions, and also the anterior cingulate cortex, which is involved in self-awareness and regulating our responses.
Wow. Okay, that really resonates with the difficulties people with depression often describe, like trouble concentrating, making decisions, feeling overwhelmed by emotions.
It does seem to map onto those experiences.
And is there a link between how much these areas shrink and the depression itself? Like, is it worse if the depression is more severe?
Yeah, the evidence suggests that. The severity and how long the depression lasts seem to be linked to how much volume reduction you see. Even how long someone's been in treatment can play a role.
So depression can literally leave a physical mark on the brain over time.
It appears so. It really underscores the biological impact.
That's quite something. But you mentioned something about adolescence being different.
Ah, yes.
Yeah.
That's a really interesting finding. Some studies have actually found greater gray matter volume in a specific part of the prefrontal cortex, the dorsolateral part, involved in planning and working memory in teens with depression compared to those without.
Greater volume. That seems counterintuitive.
It does. But one theory is that this might actually be the brain trying to compensate, like a neuroplastic response, trying to push back against the effects of the depression.
So the brain trying to adapt even under duress.
Potentially, yes. It shows that inherent drive to adjust.
Okay. Let's go back to the hippocampus shrinking. What's thought to be causing that specifically?
Well, the thinking is it might be due to disruption, maybe even atrophy, like a withering of the neurons themselves, and also those glial support cells within the hippocampus.
So the cells are actually getting damaged or shrinking.
Possibly. And crucially, this seems tied to changes in synaptic plasticity.
Right, the connections between cells.
Exactly. That fundamental ability of synapses to strengthen or weaken, which is the basis of learning and memory. If that's disrupted, it could contribute to the volume loss.
Okay, so it's not just size, it's the communication network getting hit too. Now, what about neurogenesis? I've heard that term, the brain making new cells.
Right. Neurogenesis, particularly in a part of the hippocampus called the dentate gyrus. This is one of the few spots in the adult brain where we know new neurons are continuously born.
And depression affects this.
Research, especially in animal models showing depression-like behaviors, indicates that neurogenesis is impaired there. It slows down.
Why? What puts the brakes on it?
A major factor seems to be high levels of stress hormones, glucocorticoids. We often see elevated levels in people with depression.
And stress hormones are bad for new neurons.
They can be, yes. They seem to inhibit the birth of these new cells in the dentate gyrus. It's interesting, in animal studies, if you remove the adrenal glands, which make these hormones, neurogenesis can actually increase.
Wow. That really highlights the powerful link between stress and the brain's ability to, well, renew itself.
Absolutely.
Okay, you mentioned glial cells earlier, the support crew. We focus a lot on neurons, but what role do glial cells play in these plasticity changes in depression?
They're way more than just support. Glial cells like microglia and astrocytes are active participants. They help shape brain circuits and influence plasticity. Let's take microglia first.
Okay.
These are kind of the brain's immune cells. Chronic stress, which we know is tightly linked to depression, can lead to their dysregulation.
So the brain's own defense system goes awry.
Something like that. Post-mortem studies of people with depression who died by suicide have found signs of increased microglial activity, particularly in the cingulate cortex.
And what happens when they're overactive?
Well, if they're not functioning right, they can disrupt the balance of brain networks. They can impair plasticity by releasing inflammatory molecules called cytokines, and also by messing with how synapses are pruned or remodeled.
It sounds like it could create a negative cycle. Stress triggers inflammation via microglia, and that inflammation makes it harder for the brain to adapt.
That's a real concern. There's even evidence suggesting that inflammation early in life might alter how microglia function long-term, affecting their ability to prune synapses correctly later on, possibly contributing to adolescent depression.
So early life events matter here too.
It seems so. And experimentally, if you activate microglia in mice using certain immune signals, it can impair adult neurogenesis in the hippocampus and cause depression-like behaviors. But if you inhibit that microglial activation, you might prevent those effects.
So managing inflammation could be key. What about the other type you mentioned, astrocytes?
Astrocytes are star-shaped cells with many jobs. They're increasingly seen as crucial players in synaptic function, and that function can go wrong in depression.
How so?
In animal models of chronic stress, astrocytes in the prefrontal cortex actually show signs of their fine processes, the little branches that interact with synapses shrinking back.
They physically withdraw.
That seems to be the case. Also, they store energy as glycogen, and studies have found decreased glycogen in hippocampal astrocytes linked to chronic stress and depression-like behavior. Maybe an energy deficit contributes?
An energy crisis at the cellular level.
Possibly. And in humans, studies have reported fewer astrocytes or astrocytes not working properly in those key frontal limbic circuits in people with MDD and suicide victims. This could lead to wonky synaptic connections and imbalances in neurotransmitters.
It's like the whole support system is under strain. And the third type, oligodendrocytes, they make myelin, right? The insulation on the brain's wiring.
Exactly. Oligodendrocytes form the myelin sheath, which is essential for white matter integrity, making sure signals travel quickly and efficiently between brain regions. And problems with white matter integrity have been linked to things like depression.
So the actual wiring could be affected.
Potentially, yes. Animal models show reduced myelin and related proteins in depression. But encouragingly, antidepressant treatment can sometimes reverse these changes. It also looks like problems with the development and maturation of oligodendrocytes might play a role.
Interesting.
And one study hinted that these myelination changes might be particularly noticeable in adolescent girls with depression.
Another potential difference. Okay, this is incredibly detailed, but let's try to connect it back to the synapses, the actual connections. How does all this impact synaptic plasticity?
Right. Synaptic plasticity is where the rubber meets the road, so to speak. In depression, we see changes in the structure of dendritic spines.
Spines, little bumps on the neurons.
Exactly. That's where most excitatory connections happen. Their shape, size, density, all that can change. Forming new synapses, new spines, it's linked to spine shape. You have thin, dynamic spines and more stable, mushroom-shaped ones.
And what happens in depression? Do we lose the stable ones, gain unstable ones?
It's complex, and honestly, it can vary depending on the brain region. Animal studies show different patterns, like maybe more stubby spines in the amygdala under stress, but an overall reduction in total spine density in the prefrontal cortex.
A reduction in connections in the thinking part of the brain.
Generally, yes. That seems to be a common finding, decreased synaptic and spine density in areas like the PFC and depression. And importantly, antidepressants often help reverse this loss.
That's hopeful.
It is. We also see lower levels of proteins essential for synaptic function, like synaptophysin in the hippocampus of depressed animals. And remember the microglia. They seem to be involved in altering these spines too.
So it's hitting the connections from multiple angles. And what about the chemical messengers themselves, the neurotransmitters? How do they fit into the plasticity picture?
Well, the classic view of depression involves impaired neurotransmission, particularly serotonin, norepinephrine, and dopamine. That's well documented in models.
The monoamines.
Right. But we also see changes elsewhere. Enhanced glutamate signaling via certain receptors at reduced levels of GABA, the main inhibitory or calming neurotransmitter. These imbalances definitely impact how synapses work and their ability to change.
It all ties together. Okay, and finally, neurotrophic factors, brain fertilizer, essentially.
That's a good analogy. These proteins, like BDNF and NGF, are vital for neuron survival, growth, and plasticity. And guess what? Lower levels in the blood are common in depressed individuals and animal models.
And treatments help?
Often, yes. Successful antidepressant treatment frequently leads to increased BDNF levels. It's thought that altered levels of BDNF and NGF are a key reason why neuroplasticity is impaired in depression.
So it's not just the structure, but the stuff that supports the structure and growth that's lacking. BDNF comes up a lot. What does it specifically do for plasticity?
BDNF is huge for this. It promotes spine formation, helps dendrites, the branches of neurons, grow out, and strengthens synaptic connections. If you add BDNF in lab settings, you can see these effects. It helps trigger long-term potentiation, that strengthening process essential for learning.
And lower levels are seen in depression.
Yes, including conditions like postpartum depression.
What about NGF, nerve growth factor?
NGF is also important. It's involved in neurogenesis and hippocampal plasticity. Dysregulation of NGF is linked to depression. Antidepressant treatment can help restore its levels, and interestingly, NGF itself might have antidepressant effects.
So boosting these factors seems like a promising strategy.
It's definitely a focus of research.
Okay, we've covered a lot at the cellular and molecular level. How does this translate to changes in those key brain regions we mentioned earlier? Let's start with the hippocampus again.
Right, the hippocampus. Crucial for emotion, learning, memory heavily studied, stress, a big depression trigger, induces all sorts of changes there. Volume, neurogenesis, even cell death or survival signal.
And how it communicates changes too.
Yes. Resting state fMRI studies show increased functional connectivity, basically. More communication between the hippocampus and a part of the prefrontal cortex called the subgenual ACC in depression.
So they're talking too much, maybe?
Possibly in an unhelpful way. And ketamine, that fast-acting antidepressant, can blunt this increased connectivity. Also, the hippocampus tends to be hyperactive in response to negative things in depression, and that hyperactivity can normalize after successful treatment.
So treatment helps calm it down and maybe makes its communication more appropriate. What about the prefrontal cortex, the PFC, our executive control center?
Absolutely key for thinking and behavior regulation, and definitely involved in depression. Some studies show reduced overall connectivity of the PFC with the rest of the brain, and again, ketamine seems to help reverse that.
And within the PFC, different parts act differently.
Yes. Often we see hyperactivity in the ventromedial PFC, VMPFC, and hypoactivity, less activity, in the dorsolateral PFC, DLPFC. But treatments, whether therapy or meds, can help reverse this imbalance. And crucially for plasticity, impaired neuroplasticity has been found specifically in the DLPFC in depressed individuals.
So the part responsible for flexible thinking loses some flexibility itself.
That seems to be the implication.
Okay, and the amygdala, the emotion center.
The amygdala also shows altered connectivity, reduced communication with the orbitofrontal cortex, which processes rewards. Maybe lower connectivity within the amygdala itself if anxiety is also present.
But sometimes increased connections?
Sometimes, yes. Increased functional connectivity between the amygdala and the right PFC has been reported, which can then decrease after SSRI treatment. It's complex, likely depends on the type of depression severity, maybe when it started.
It really shows depression isn't monolithic. What about the reward system, the ventral striatum? Linked to anhedonia, right? That lack of pleasure.
Exactly. The nucleus accumbens within the ventral striatum is key here. Lower activation in this area is a consistent finding in MDD. But the good news is, this often normalizes with effective treatment. The reward circuitry seems capable of bouncing back.
That's encouraging. And lastly, the lateral habenula, the anti-reward center.
Right, the LHB. It puts the brakes on motivation, responds to negative outcomes. Its dysfunction is increasingly implicated in MDD. A key finding is increased excitatory signaling onto LHB neurons that project to the VTA, a dopamine area. This hyperexcitability might be a cellular basis for some MDD symptoms.
Fascinating how specific circuits can become over or underactive. Okay, so we've painted a picture of how depression affects brain plasticity. Now the crucial part, how can treatments help turn things around? Let's start with conventional antidepressants.
Okay, conventional antidepressants, the TCAs, SSRIs, SNRIs, they were mostly developed based on the monoamine hypothesis, targeting serotonin and norepinephrine. But we now know their effects go beyond just boosting those chemicals short term.
Right, you mentioned plasticity effects.
Yes. Chronic long-term use seems to enhance synaptic plasticity. They can promote hippocampal neurogenesis, increase those neurotrophic factors like BDNF, and encourage synapse formation. So they appear to help the brain rebuild and adapt over time.
Let's break them down. TCAs, the older ones.
Tricyclics. They do increase serotonin and/or norepinephrine. Postmortem studies showed more neural progenitor cells that become neurons in the hippocampus of treated patients. Animal models show they promote cell proliferation there too. But they have side effects because they hit other receptors, so they're used less now.
Then SSRIs, the common ones like Prozac, Zoloft.
Serotonin selective reuptake inhibitors. Much more targeted, generally fewer side effects, often first-line treatment. Animal models show they boost BDNF in astrocytes, promote synaptogenesis, enhance neurogenesis. They definitely impact plasticity.
Still have side effects for some people, though.
They can, yes. And there's some complex data suggesting maybe in some specific contexts, certain SSRIs might paradoxically hinder plasticity, but that needs more research. Mostly the evidence points towards promoting plasticity.
And SNRIs, like Effexor, affecting both serotonin and noradrenaline.
Serotonin and norepinephrine reuptake inhibitors. Similar story to SSRIs in terms of promoting neurogenesis and plasticity in animal models. Venlafaxine, for example, can help prevent stress from damaging BDNF levels and hippocampal neurogenesis.
So these established meds seem to work at least partly by fostering the brain's own repair mechanisms over the long term. But what about the faster-acting ones? Ketamine generated a lot of buzz.
It really did. Ketamine, an NMDA receptor antagonist, can have antidepressant effects within hours, and it seems to work partly by rapidly boosting plasticity. It enhances synapse formation, restores lost spines in animal models.
Yeah, and helps brain networks communicate better.
Yes, it can help normalize connectivity, for instance, increasing connectivity between the frontal cortex and the striatum in treatment-resistant depression. It's a very different mechanism.
Are there others in that rapid-acting category?
Yes, several are being explored. Rapastanol, another NMDA modulator, showed promise in animals by enhancing plasticity, but unfortunately it didn't pan out in large human trials.
Shame.
Scopolamine, which blocks muscarinic receptors, also shows rapid effects, maybe by promoting synaptogenesis and boosting glutamate transmission in the PFC, but durability and side effects are concerns.
And psychedelics, psilocybin, LSD.
Growing interest there. They seem to promote rapid structural plasticity, helping neurons grow branches and spines, form synapses, particularly in the frontal cortex, potentially through BDNF pathways. Still early days for clinical use, but promising findings on plasticity.
Very different approaches targeting plasticity. What about melatonin-related drugs affecting sleep cycles?
Melatonin receptor agonists, MRAs. Galatine is one used for MDD. It acts on melatonin receptors, but also blocks certain serotonin receptors. It's shown to upregulate BDNF.
Interesting combination.
Ramotine might help older adults with depression and insomnia, possibly by normalizing circadian genes and neurotrophic factors. And new EP11, or pyromelatine, is a newer one that also seems to enhance GABA, the calming neurotransmitter, and boost BDNS in the hippocampus.
So even regulating sleep-wake signals can influence plasticity factors. Okay, let's switch gears to direct brain stimulation. ECT, TMS.
Right. Brain stimulation directly modulates brain activity and plasticity. ECT, electroconvulsive therapy, despite its reputation, it's highly effective for severe resistant depression.
And it causes brain changes.
Big time. It induces structural changes, increases in gray and white matter volume. It promotes synaptogenesis, neurogenesis, gliogenesis, basically growth across the board. It's a powerful plasticity inducer, though it can have temporary cognitive side effects.
What about less invasive options, TMS?
Repetitive transcranial magnetic stimulation, or TMS, uses magnetic pulses non-invasively, modulates cortical activity and connectivity, shown to have antidepressant effects, likely via plasticity mechanisms.
And vagus nerve stimulation.
VNS, and its non-invasive cousin, TVNS, targeting the ear. They can regulate brain network activity, alter NGF and BDNF levels. TVNS is particularly interesting as it doesn't require surgery.
What about DBS and TDCS?
Deep brain stimulation, DBS, involves implants, targets specific circuits, and promotes neurogenesis and plasticity by altering gene expression. Transcranial direct current stimulation, TDCS, uses weak electrical currents, non-invasively, thought to influence brain function by promoting plasticity. It's being explored for MDD treatment.
So a whole toolkit of ways to directly nudge the brain towards plasticity. What about non-medical approaches? Therapy, exercise?
Absolutely crucial. CBT, cognitive behavioral therapy, a first-line psychotherapy, doesn't just change thoughts and behaviors. It's been shown to reverse volume reductions in the hippocampus and nucleus accumbens and normalize connectivity in reward circuits. Therapy changes the brain.
That's powerful.
And even simpler things, learning positive emotional strategies can enhance synaptic plasticity in animal models. Exercise is huge.
Oh, so?
Running, swimming, they improve mood, yes, but also normalize brain activity patterns, increase plasticity-related proteins, boost neurogenesis in animal studies. It's a potent brain health intervention.
Oh, yoga.
Some studies suggest yoga might increase GABA levels in the brain in depressed individuals, potentially contributing to calming effects and plasticity. Even lactate produced during exercise might enhance hippocampal neurogenesis.
It's amazing how lifestyle factors directly impact brain biology. And quickly, you mentioned other research areas, genetics, traditional medicine.
Yeah, briefly. Gene modulation studies in animals are identifying specific genes that influence depression-like behaviors and synaptic plasticity, potential future drug targets. And various compounds from traditional Chinese medicine and herbs like curcumin or ginseng show antidepressant-like effects in animal models, often linked to improving plasticity, reducing inflammation, boosting neurotrophic factors.
A lot of avenues being explored.
Definitely. Plus, other compounds, even things like statins or lithium, show potential effects on neuroplasticity relevant to depression in preclinical studies. The field is constantly looking for new ways to leverage the brain's ability to change.
Okay, this has been incredibly comprehensive. We've gone from brain cells shrinking to how treatments like exercise or brain stimulation can help rebuild connections. To wrap things up, what are the main takeaways for people listening?
I think the biggest takeaway is that depression isn't just a mood thing. It's associated with real, measurable changes in the brain's structure, function, and its fundamental ability to adapt its plasticity.
Right. Changes in key areas for mood, memory, thinking.
Exactly. Affecting cell growth, connections, chemical signaling, support systems. It's complex and affects the brain on multiple levels.
And this feels like the most important point is not a fixed state, is it?
Absolutely not. That's the crucial message of hope. The brain is plastic. And a whole range of interventions, medications new and old, therapies, brain stimulation, even lifestyle changes like exercise, demonstrate a remarkable ability to positively influence that plasticity. They can help the brain heal and rewire itself.
Recovery is biologically possible.
Yes. The brain has an inherent capacity for change.
And science keeps uncovering more about how this works.
Constantly. We're learning more about the mechanisms, identifying new targets. It really emphasizes how dynamic and adaptable our brains are. Even in the face of something like depression, the potential for developing even better, more personalized treatments in the future is very real.
Which leads to a final thought, maybe. If the brain can change in so many ways, how might we tailor these approaches even better in the future? What personalized strategies could unlock more effective, more sustainable paths to recovery for each unique individual? Something to think about.
Definitely food for thought.
Thank you so much for walking us through all of that. It's been really enlightening.